So because of pandemic, people's interest in the study on social interactions of social interactions of human communities, but humans are not the only social animals. And in this talk, I'd like to talk about the universal patterns of social behavior that can be observed in both humans and non-human living beings and the implications of it. So I want to first point out that a community is more than a collection of individuals. So in this picture, there are a bunch of individuals gathered on the quad at UIUC before the pandemic, of course. And these people are not just coexisting there, but they are interacting with each other. And this interaction gives rise to the collective property that characterizes a community. Social network describes how this interaction happens. So to study this emergent collective property, we look at the social network. The network structure shows how the community is connected inside, and it often shows the shared patterns of social behavior across different systems. So for example, this picture shows the degree distribution. So degree is the number of connections each individuals have plotted in log scale. And all these four cases are different communities, but they are all poorly distributed. This means that although the context is different, all of them share the pattern of behavior such that there are lots of individuals with small number of connections and small number of individuals with lots of connections. And figuring out these patterns of behavior, we can study the topics that we are actually interested in, such as the spreading of disease or information. So this picture shows basically how the disease spreads within the community. And you can see that this individual, for example, has a larger contribution to the disease spreading compared to like this individual, for example, because this person has more friends. And how information spreads is just like how the disease spreads. So people use the same formalism to study the spreading of fake news or internet memes, so on the internet. Here is another example of pattern of behavior. So this is how the students interact with each other. And you can see that students tend to interact more with their classmates compared to students in other classes. So by realizing these patterns of behavior, we can use this to come up with the strategies to slow down the disease spreading or like fake news spreading, etc. As I said earlier, humans are not the only social animals. There are many other living beings such as honeybees, whales, and even bacteria, and many more other cases that form a community, and their social interaction is vital for their survival. So one may be curious whether we can find the universal and cross-species patterns of social behavior we can see in both human and non-human living beings. And if we can find those, what do they tell us about the very nature of social interactions? And to answer these questions, we first need a high-throughput technique to track the social interactions. So now I'm going to tell you how my experimental collaborators monitored the honeybee social interaction. So honeybee was our model system for non-human animal because they're quite convenient. There are thousands of individuals in a colony, which means that we can get lots of data per colony, and they are relatively easier to experimentally manipulate. There are many different kinds of honeybee social interaction, but we chose Trophallaxis, which is mouth-to-mouth liquid food transfer. And it happens not only for feeding, but also for communicating. And here I'm showing you the two honeybees doing Trophallaxis, and you can see that when they are Trophallaxing, they are actually physically connected through their proboscis. So we can easily tell whether they are actually interacting with each other or they are just sitting next to each other doing nothing. Here's our experimental setup. The honeybee colony is located in this two-dimensional observation hive, And this high-resolution camera images the colony at the rate of 1 frame per second. And we tracked 5 colonies, and each of the colony had about 1,000 honeybees, and we observed them for their entire lifetime. Each honeybee was attached with this barcode, and this barcode has unique ID with it. So we can track which honeybee interacted with which honeybee and how long they interacted and when they interacted, etc. And as I said earlier, we can actually tell whether or not they are doing Trophallaxis. There are many kinds of patterns of interaction, and a common quantity that people choose to quantify the temporal pattern of interaction is inter-event time distribution. So inter-event time is the time between two successive interaction events. So in this picture, this white band means nothing happened, and this vertical line means that the interaction happened at that time. So there are there can be many different scenarios. So the interaction can happen periodically with some noise. So in that case, the inter-event time distribution would be Gaussian. If the interaction happens in a Poissonian way, the distribution would be exponential. And if the interaction happens intermittently, meaning that there are long periods of nothing happening, followed by a relatively short period of time where lots of interaction happens, followed by another long period of nothing happening, so on. So this pattern is called bursty, and in this case, the distribution is polar. So unlike other two cases, bursty pattern does not have a characteristic time. For a human case, the interaction pattern is actually bursty. So this is the inter-event time distribution plotted in log scale for the email correspondence. And you see this linear line. So since this is log, this means polar distribution. And it is thought that the burstiness is responsible for slow spreading of information or diseases. So in our lab, the previous graduate student measured the inter-event time distribution for the honeybees, and this is the result plotted in log scale again. And you see this linear line, which means that just like humans, honeybees also have a polar inter-event time distribution, so they have bursty interaction pattern. So other possible question is, what about the duration of the Trophallaxis itself rather than the time between two events? So that was the question I wanted to answer in my study. So I used the same experimental data that was used for the inter-event time study. And here is the experimental result. So it is the duration, the distribution of the duration of Trophallaxis plotted in log scale again. And these are all different colonies. And you can see that all of them show polar distribution, and these are the exponents for the power law. So I wanted to come up with a theory to explain why you would see a polar distribution of the duration of the interaction. So to answer that, I viewed the bond between a pair as a particle with two states representing an interacting pair and non-interacting pair. And this particle changes its state by jumping over the energy barrier with height e with rate omega. So the time it takes for this particle to jump over the energy barrier is the duration of the interaction, or what I call interaction time in this work. And for a single barrier height case, the interaction time distribution is exponential, and that can be calculated by multiplying the probability not to jump until time t with the jumping rate omega. But it turns out that some pairs tend to interact longer than others, and that longer interaction can be achieved by having a larger barrier height, because it is harder for the particle to jump. So this variability in the population can be represented by different barrier heights with the distribution P of E. So the natural question is, what is the function of form of P of E? So to answer that, I thought of a scenario where this center P wants to interact in the next time step, and there are neighbors, and these neighbors are the candidate partners. And with each candidate partners, it will have different value of energy barriers. And the B pair with the highest energy barrier height would be the stablest because, again, if the barrier height is higher, it is harder for the particle to jump. So it is harder for the pair to break. So we assumed that this pair with the highest energy barrier height would be the one we end up observing in our experiment. In other words, the B pairs we see in our experiment are the collection of the B pairs with the maximum energy barrier heights. So P of E would be the extreme value distribution for maxima. In case you are not familiar with the extreme value statistics, I'd like to just briefly explain what it is. So P of X is some random probability distribution, and we pick independent and identically distributed random variables from this distribution. So we pick one batch of the numbers, and after that, independently, we pick another bunch of numbers. So that's the second batch. We pick third batch, fourth batch, etc. And in each batch, there will be some maximum number. So for example, among these numbers, 0.51 is the largest. Among this batch, 2.2 is the largest, etc. So we can collect the maximum number from each of the batch, and the distribution of these maximums is the extreme value distribution. Depending on the tail behavior of the parent distribution P of X, the extreme value distribution F of Z may converge to one of the three cases. So if P of X decays faster than power law, then F of Z would converge to a Fisher-Tippett-Gumbel distribution shown here. If P of X decays like this, F of Z will converge to Frechet distribution, and if P of X decays like this, F of Z would converge to Weibull distribution. In the nature, Fisher-Tippett-Gumbel distribution is the most common one, so we assume that our extreme value distribution would be also Fisher-Tippett-Gumbel distribution. And we are only interested in the tail behavior of our interaction time distribution, so that's the long time limit. So we took the large E limit, then this super exponential term will go away, and this asymptotic behavior of P of E is the extreme exponential distribution. So we have this exponential pair interaction time distribution, exponential P of E, the barrier height distribution, and by using the Cramer's rate formula, we can calculate the population interaction time distribution, which I will call F of T from now on, by integrating this pair interaction time distribution over energy barriers. And the asymptotic behavior at long time is a power law, and that is what we see in our experiment. Note that the exponent in the power law of F of T is related to the exponent in the barrier height distribution. As a side note, it turns out that this exact same calculation has been done in the literature of glass. But in the glass case, one atom hops over multiple energy barriers to escape from this potential well. But yeah, so that's the integration over the energy barriers is the average over successive hops experienced by one atom. On the other hand, in our social interaction case, each particle jumps only once, but we are averaging over the population. So that's ensemble average over the population. So although the mathematical equation is the same, the interpretation is different. So back to our theory, we have reproduced what we see in the experiment. So we should now verify our theory actually makes sense by verifying the predictions made by theory. The first prediction is the exponential pair interaction time distribution. To verify that, we plotted the quantile plot of pair interaction times. So quanti-quanti plot is basically comparing the cumulative distribution of the data and the proposed function. And if the data follows the proposed function, the data point will lie on this linear reference line. In our case, it lies well up to some point and deviates upward. But if you look at the deviating part, even the deviating part is actually linear. So we conclude that pair interaction time distribution is actually better described by a weighted sum of two exponentials with different rates rather than a single exponential. So that's called hyperexponential function. And quite fortunately, our theory is not affected by this modification. So the calculation for the population interaction time is now double integral with this hyperexponential pair interaction time distribution. But the asymptotic behavior at large t is again a polar with the same exponent. So that was the result for just one pair, but as I said earlier, there are like thousands of honeybees, and that means there are like a larger number of pairs, and we cannot check like each pair by hand. So we wanted to check whether this hyperexponential function is a good fit for the pair interaction time distribution for all the pairs in our data at once. So we use data collapse, and just like before, if hyperexponential function is a good fit, the data point should lie on this reference line, and you can see that it is quite a good fit for all the pairs in all the colonies. So what this like hyperexponential distribution tells us is that there are actually two energy barriers, not just one, and each energy barrier corresponds to each exponential term. And we think that that could be because the interaction can be terminated by either this right honeybee or left honeybee. So, for example, if the interaction was terminated by this right honeybee, the particle weight will take this path to jump to this other potential well. But if the interaction was terminated by this left honeybee, then the particle will take the other path to jump to the other potential well. So we have verified this first prediction with some modification. That doesn't change our central result. The second prediction is the exponential energy barrier height distribution. But energy barrier height is not something we can measure directly, so we used Kramer's rate formula again to transform P of E into P of tau, where tau is the mean pair interaction time. Note that this P of tau is also polar with the same exponent as f of t. So by comparing P of tau and f of t, we can verify our second prediction. Before I go further, I'd like to just be extra clear about f of t and P of tau. So here we have three honeybee pairs, and each bee pair have multiple interaction events, and the duration of each interaction event is t. So we collect the duration of all the interaction events by all the pairs and get the distribution, and that's f of t. On the other hand, mean pair interaction time tau is, as the name suggests, the mean value of the interaction times between a pair. But we have two taus rather than one tau per pair because there are two energy barriers per pair. So we have to get tau associated with each energy barriers. And so we get these two taus per pair and collect the taus and get the distribution, and that's P of tau. Here's the comparison of f of t and P of tau in plotted in log-log scale, and you can see that they have the same tail behavior. So this red one is f of t, this blue cross one is P of tau. And I'm only showing you the result of this one colony, but we basically see the same pattern for all the colonies being analyzed. So both predictions have been verified. So I showed you that for honeybees, the interaction time distribution is heavy-tailed, which means power law, and pair interaction time distribution is hyper-exponential, and population interaction time distribution f of t, and mean pair interaction time distribution p of tau have the same scaling. Nice thing about our theory is that it is general enough that it can be applied to not only honeybees, but also other systems such as humans. So we got the human face-to-face interaction data from the social parents collaboration. So you can also get their data by visiting their website here. And we plotted the duration of these human face-to-face interactions in log scale again, and you can see that for all these different contexts, such as school, hospital, conference, etc., they are all parallel distributed, just like honeybees. And we also checked whether the pair interaction time distribution is hyper-exponential function for humans as well by using this data class to see the result at once again. And you can see that it is also a good fit for humans as well. And we compared the scaling of f of t and p of tau, and you can see that they have the same tail behavior. And again, I'm only showing you just one data set from humans, but we see the same scaling for all the data sets. So this means that for humans, the energy barrier height distribution is also exponential. So here is the comparison of the scaling for all the data sets we analyzed. So for these above ones are honeybees, and these below ones are humans. So these red data points are population interaction time, f of t, and you may wonder why I'm showing you two different kinds of mean pair interaction time distribution, p of tau. I have already told you that since we have two energy barriers, there should be two taus per pair. But if we actually look at the data, they are just numbers. So we cannot tell which interaction event was associated with which energy barriers. So to get this tau, we have to actually fit the pair interaction time distribution to this hyperexponential function and get the taus by getting the reciprocal of the fitting parameter omega. So here, the subscript I just labels the pair, and alpha labels different energy barriers. But fitting is quite burdensome, so one may want to just ignore all this like two energy barrier height stuff and just get a single easy average of all pair interaction times. So I call this tau I because I is labeling the pair. And this tau I will be the linear combination of the tau I alpha. One may ask whether it matters which one I use. So from now on, just for convenience, I'll call this tau average and this tau fit. So it turns out that it didn't matter for most of the data sets, but it mattered for these two particular data sets. So let's zoom in on one of them. So again, this red one are f of t, and the blue cross one is tau fit, and the black one is tau averaging. And it's quite clear that tau fit and f of t and p of tau fit have the same scaling, but p of tau average is deviating. And if we look at the exponent of f of t for all the data sets, it turns out that the ones where all three distributions had the same scaling had the exponent between -2 to -3, while the deviating ones had exponent -3.4 and -3.6. So it seems like the exponent matters. In other words, only the datasets with an exponent smaller than -3 show the disagreement between p of tau fit and p of tau average. So we can explain this by using the stable law. So a distribution is called stable if a normalized sum of IID random variables is also a random variable from the same distribution. So Gaussian distribution is a good example of a stable distribution. And what the stable law says is that the distribution of the normalized sums of IID random variables converges to a stable distribution. And what kind of stable distribution it converges to depends on the decay or the tail behavior of the parent distribution. So let's say the parent distribution f of x decays like x to the minus alpha plus 1. And if alpha is bigger than 2, the converging stable distribution is Gaussian. And that's the central limit theorem that we are familiar with. But if alpha is between 0 and 2, this parent distribution f of x itself is stable, so the distribution of the sum f of y has the same scaling as its parent distribution, like this. In other words, the distributions decaying faster than x to the minus 3 are not stable, so they don't have the same asymptotic scaling as the distribution of the sums. In our case, the tau fit is the parent, so P of tau fit is the parent distribution, and since tau average is the linear combination of tau fits, P of tau average is the distribution of the sum. So going back to our data, the population interaction time f of t and the distribution of tau fit, P of tau fit, always have the same scaling according to our data, and that agrees with our theory. But tau from averaging may or may not have the same scaling depending on the exponent. So the two particular ones that didn't have the same scaling, the primary school and the household data sets, so they fall into the regime where P of tau fit is not stable. So P of tau averaging does not have the same scaling of P of tau fit, and since P of tau fit and f of t always have the same scaling, P of tau average also deviates from P of f of t. All the other data sets fall into the regime where the parent distribution P of tau fit is stable, so all three distributions have the same scaling. So what this tells us is that we actually cannot ignore the fact that there are two energy barriers per pair. So although it's burdensome, we actually need to fit the pair interaction time distribution into the hyperexponential function and get the two taus per pair to get the mean pair interaction time distribution. So far, I've shown you that both honeybees and humans have power law interaction time distribution. Both of them have hyperexponential pair interaction time distribution, and both of them have the same scaling between f of t and p of tau. And the lesson from this is that the individual differences in the population, which was represented by the energy barrier height distribution, is responsible for the universal heavy tail and the universal scaling law we are seeing in our data. It is quite obvious that humans are different, but it is not so obvious for the honeybees. And in fact, only recently the individual differences in the worker honeybees has started to be explored. So we wanted to show that the worker honeybees are actually different in a way that is independent from our theory. So we measured the Gini coefficients for the total time spent for interaction, total number of interactions, and total number of partners. So Gini coefficient is a quantity used in economics to quantify the inequality in the society. So in our case, the inequality would mean the unequal contribution to the social interactions that is happening in the community. So let me just briefly tell you how to calculate the Gini coefficients. Let's say we have like completely equal society and unequal society. And to calculate the Gini coefficient, we should first order the individuals in the order of increasing wealth and get the cumulative distribution of the wealth. So for the totally equal society case, because everybody has the same wealth, this curve is linear. On the other hand, for the unequal case, the curve deviates from this linear reference line. And the Gini coefficient, G, is the ratio between the area between this linear line and the deviating curve, A, over this total area of the triangle. So for the completely equal case, because there's no deviation, the Gini coefficient G is 0. And the more unequal the society is, the closer G is to 1. And this plot is called Lorentz plot. So this is the Lorentz plot for the honeybees and humans for total time spent for interaction. So I'm showing you the results for the total time, but we basically get the same result for all the other criteria that we analyzed. So you can see that both honeybees and humans show this deviating area. That means they are unequal, and the Gini coefficient for honeybees is between 0.2 to 0.3, and for humans is between 0.3 to 0.5. So this shows that honeybees are indeed unequal, but it seems that honeybees are less unequal than humans. So they have like smaller area and smaller Gini coefficient. And we speculate that that could be related to the closer genetic relatedness of the honeybee colony, because in our experiments, the queen bee was inseminated with just one single drone, one single male, I mean. So the worker honeybees were sisters sharing the 75% of the gene. But that's just a speculation and we haven't verified. This was not the first time that we used this Gini coefficient to quantify the inequality in honeybees. So this work was done in our lab again before I even got admitted to graduate school. So the previous graduate student quantified the inequality or individual differences in the foraging activity of the honeybees. So foraging activity is the activity of collecting the food from outside. And the student plotted the Lorentz plot, and we clearly see the deviating curve, and the Gini coefficient was around 0.5. So this is another example showing that the worker honeybees indeed show individual differences. So in conclusion, both honeybees and human interaction time distribution is polar distributed, and this heavy tail arises from the individual differences in the population, and we represented these individual differences with different energy barrier heights for this particle picture, and this energy barrier height was assumed to be extreme value distributed. And although it was not obvious for worker honeybees whether they are indeed different or not. So we showed that they are indeed different in a way independent from our theory by calculating the Gini coefficient and showed that they are indeed different. So the effect of these individual differences in the population on the parents of social behavior is still open for further studies. Thank you.